Good morning children. Saira, welcome to this session. Today we are going to continue with lesson number 8, Cell Biology and Biotechnology. Already we have seen two parts of it. Let us revise what we have learned. We studied about cytology. What is cytology? A study of cell organelles, cell division. All this about cells is called as cytology, and we learnt about the stem cells. Okay, and what is the use of the stem cells we saw, and we learnt about organ donation. Okay, and we started with biotechnology, and we learnt about the benefits of biotechnology. Under that we learnt about the commercial application. Under that we saw, under first one we saw was crop biotechnology. Okay, we learnt about hybrid plants, hybrid seeds, and GM crops. Okay, golden rice. All these we learnt under this crop technology. Now let us continue. The next first one was crop technology. Biotechnology we learned how it is useful for making good crops as well as good yield and good variety of crops with the help of biotechnology. Now let us continue. Next one, biotechnology is also useful in animal husbandry. Two main methods as artificial insemination and embryo transfer are used in animal husbandry. Animal husbandry means improving the animal breed as well as the quality of the animals health. If the cattle are there, they will give a lot of milk. Okay, so there will be healthy variety of animal is got with the help of this biotechnology by the methods of artificial insemination. That means semen of artificially it is inserted into the female of the female animal. Artificially the semen of the other male is inserted, and you get an embryo will be a good quality one. Okay, and even the embryo transfer is also can be done with the help of biotechnology. It helps to improve both the quantity and quality of animal products. Okay, example, you get a lot of milk, meat, wool, etc. you get with this method. Similarly, animals with more strength have been developed for hard work. Okay, so all these methods are improving the quality as well as quantity of the animal products. So what are the two methods in animal husbandry with the help of biotechnology? Artificial insemination method as well as an embryo transfer method is used with the help of biotechnology. Understood? So what is why it is done? You get better quality of animals as well as animal products and good quality and quantity of animals also. Okay, quantity also will be more. Milk, you get lot of milk, meat, wool, etc. Okay, when you animals like ox, buffalo, etc. will have good strength when you do this method for them to do the hard work. Okay, now let us see the third one, human health. Biotechnology is also useful for human health. Already we have learnt about vaccines, antibiotics, all these we have learnt from standard 9 we are learning. Diagnosis and treatment of the disease are two important aspects of the human health management. See, suppose somebody is suffering from disease and we are not able to diagnose that disease, it is not possible to give the correct treatment. Okay, suppose a person is suffering from some major disease like cancer, but we are not able to analyze it or diagnose it, so we cannot give the treatment for that. So diagnosis and treatment are the two important aspects of the human health management. Once we diagnose the disease, it is possible to give treatment. Okay, biotechnology helps to identify the role of gene if any disease in person. Diagnosis of diabetes and heart disease has become possible even before the onset of symptoms with the help of biotechnology. So even before getting the disease like heart disease and diabetes, we can get before the symptoms start itself. We can, with the help of biotechnology, we come to know that this disease it is going to attack or it is going to affect us. So with the help of biotechnology, this can be found out. Diagnosis of the disease like AIDS, dengue can be done within few minutes. Hence, treatment can be done at the earliest. So whenever we start the treatment early, that is much better. Okay, once the disease has become chronic, it is very difficult for us to treat. Various medicines are used for the treatment of disease. Example, the hormone insulin is used to treatment of diabetes. We all know this. Earlier, insulin was being collected from the pancreas of horses. So, how insulin was used for treatment of diabetes, but how it was got? It was got collected from the pancreas of the horses. But now what it has happened? However, nowadays due to biotechnology, insulin can be prepared with the help of bacteria. This we have learnt in that previous microbiology lesson. For this purpose, human insulin gene has been inserted into the genome of bacteria. Genome means the entire DNA sequence of an organism, isn't it? Okay, so for this purpose, what has happened? Human insulin gene, okay, the small gene is being inserted into the genome of bacteria. So various vaccines and antibiotics are also produced in the same way. We learnt about vaccines and antibiotics from standard 9. We are learning about antibiotics, isn't it? So before, how it was born? It is collected from the pancreas of the horses, but nowadays is prepared with the help of bacteria. How? For this, human insulin gene has been inserted into the genome of the bacteria. Then various vaccines and antibiotics are also produced in the same way. Okay. Vaccines and vaccination. Vaccine is what is vaccine? Vaccine is the antigen containing material given to acquire either permanent or temporary immunity against a specific pathogen or disease. Traditionally, vaccines were prepared with the help of pathogens. Completely or partially killed pathogens were used as vaccines. Okay. So what is vaccine? Vaccine and vaccine. Vaccine is what is vaccine? It is the antigen containing material given to acquire either permanent or temporary immunity against a specific pathogen or disease. Okay. How traditionally, how it is prepared with the help of pathogens, real pathogen. Pathogens means bacteria or microorganisms which cause diseases, isn't it? So how vaccines were prepared before? With the help of pathogens. Completely or partially killed pathogens were used as vaccines. What was used? Either partially killed or completely killed pathogens were used as vaccines. But now how we are using? Okay, and however, due to this, there were chances of contracting the disease in some case, in some person, because it is partially killed pathogens, so it can cause disease in the person. But as an alternative, scientists try to artificially produce vaccines with the help of biotechnology. So instead of using the whole pathogen, okay, for producing vaccine, they made artificially produced vaccines with the help of biotechnology. For this purpose, scientists produce the antigen in laboratory. Okay, what did they do? They produce the antigen in the laboratory with the help of gene isolated from the pathogen and used as a. So for this purpose, what did they do? They produce the antigen in laboratory with the help of the gene isolate, only with the gene, not the whole pathogen. Okay, with the help of gene isolated from the pathogen and used as vaccine. The safer vaccine are produced. It is produced in the laboratory with the help of only the gene of the pathogen, not the whole pathogen. Okay. Now proteins which acts as antigen are injected in pure form instead of injecting the killed or semi-killed pathogen. Before, what they were using? Killed or semi-killed, half partially killed or fully killed pathogens were used. But now what they are using? Proteins which act as antigen are proteins which act as antigen are injected in the pure form, okay, instead of injecting the killed or semi-killed pathogens. These proteins keep the person away from the disease by keeping the immune system active. Thus, injecting the antigens is safest way of vaccination. So, which is the safest way? Injecting the antigen. Okay. So, the body will produce the antibodies and then they will fight against the disease. So, vaccines produced with the help of biotechnology. Why it is used according to biotechnology? They are more thermo-stable and remain active for longer duration. Why it is used, which is much preferred with the help of biotechnology? Because they are more thermostable and remain active for longer duration. So vaccines of polio, hepatitis, see almost polio is eradicated now because of this new biotechnology method of producing, of injecting antigen. Okay. So vaccines of polio, hepatitis are produced by this method. Okay. Now this part we are going to learn about the edible vaccine. There are vaccines which are called as edible vaccine. What is meant by edible means the thing you can eat it. Okay. So what is meant by edible vaccine? Work on production of edible vaccine is in progress and presently potatoes are being produced with the help of biotechnology. You can see with the help of biotechnology even edible vaccine, that means the vaccine which can be eaten, can be produced. These potatoes are called as transgenic potatoes. What are these potatoes called? They are called as transgenic potatoes. These potatoes will act against bacteria like Vibrio, cholerae, E. coli. Okay. Consumption of these raw potatoes generates the immunity against cholera and the disease due to E. coli. What will happen if these potatoes are cooked for consumption? When you cook it also, the amount of proteins will reduce. So, it is eaten as such. Okay. So, this is edible vaccine which is done with the help of biotechnology and they are called as transgenic potatoes. So, they are helpful against the diseases which are caused due to Vibrio cholerae, for disease like cholera as well as E. coli. That is also a disease. So this vaccine which is called as edible vaccine, it is made with the help of the potatoes which is called as the transgenic potatoes. You can see the picture of it. You can see the isolation of desired gene from human. From the human pathogen, they take the isolated, the gene is taken and it is inserted into the potato leaves. Okay. And this potato, each, we learnt about tissue culture. From each cell also new plants can come. From each tissue also new plants can come. So this, they are inserted into the leaves of the potato and it is made it into pieces, and this virus is left into the pieces of the leaves and it infects the leaves, and from that leaves there is the growth of the plant. So when the plant grows, even every part, entire plant is developed from the leaf pieces containing genes of human pathogens. So whichever disease pathogen is inserted, that type of genome. So consumption of this raw potatoes helps to develop immunity against that pathogen. It is like an antibiotic. Okay. So it causes immunity against that pathogen. So this is transgenic potatoes. Okay. Now next one, it is treatment. We saw about antigen vaccines and this treatment. Biotechnology is useful for production of hormones. We saw this. Insulin, somatotropin and blood clotting factors. So what is the treatment? So for treatment, biotechnology is useful for production of hormones like what all? Insulin, somatotropin and blood clotting factors. Next one, interferon. This is a group of small size protein molecule used in treatment of viral disease. What is interferon? It is a group of small size protein. It is a protein. Interferon is a protein molecule used in the treatment of viral diseases. Interferon, viral disease. Remember like that. Interferon, treatment of viral disease. These are produced in blood. So, however, nowadays with the help of biotechnology, transgenic E. coli are used for production of interferons. With the help of transgenic E. coli, that is a microorganism, are used for the production of interferon. Interferon is used for what? Viral disease, treatment of viral disease. Okay, see the proteins produced by biotechnology and the names of disease that are used again. This is inside the table. You can see it proteins produced by biotechnology. One is insulin, which is useful for treatment of diabetes, somatostatin dwarfism, erythropoietin, anemia, and the protein is factor VIII, hemophilia, okay, interleukin, cancer, interferon, viral infection. We learned this, interferon viral infection. So insulin, the proteins produced by biotechnology. One learn this table, children. It can come for fill in the blanks, so much the following. Okay, I'm not sure whether it is deleted, but better learn this because it is an important point. Okay, see the proteins produced by biotechnology. Insulin is produced. The proteins are insulin, and it is useful for diseases. It is cure disease, diabetes. Somatostatin for the disease dwarfism. Erythropoietin for anemia. Factor VIII for the disease hemophilia. Interleukin for cancer. Interferon for viral infection. So it can come even for match the following. So better learn this properly. Okay, children. So this much we have done today. We will continue with cloning. Okay, we will continue from cloning in the next video. Hope you all understood. This is how biotechnology is useful for antigen, production of antigen, and for transgenic edible vaccines, treatment. All these we have learned today. How biotechnology is useful. We will continue this in this lesson in the next PPT, and after finishing this full lesson, I will revise this lesson once again so that it becomes easy for you. Okay, children. Thank you. Sai Ram, children. Take care.